All right, so we want to run through that very quickly, that warm-up today with you. Again, these, you should have this 100% filled in because we asked you to pull out your resources. We asked you to pull out your guided notes from section 1, and it talks about the 4 reasons why cell divide in section 1. We asked you to pull out your POGL, and that for sure has every single part of the 4 phases, as well as your guided notes. I don't remember if POGL has the whys, but 4 reasons why a cell will divide or go through the cell cycle, which is the division process, is so the cell can, or your body can repair old or damaged cells. Old cells, I guess we should really say. Repair body. No, let me say repair. Damaged, right? So repair. What else? To grow. Okay? To grow. You are physically taller today than you were 3 years ago because the cell cycle has occurred in your bones.
Yep.
Okay? What else? Okay.
Reproduction.
Reproduction. There's 2 types of reproduction: sexual reproduction and asexual reproduction. Reproduction through the process of mitosis is called asexual reproduction. We'll get into this a little bit more later, where one organism divides to make 2 organisms. Now, through the cell cycle, asexual reproduction is where one unicellular organism divides to make 2 unicellular organisms. Okay. Also, the making of what we call sex cells, egg and sperm cells, which combine during sexual reproduction, are also made during the cell cycle. It's just that M phase is not mitosis now, it's meiosis, but we'll get to that.
We'll get to that.
And last,
Replacing old cells.
to replace those cells. Now, in my mind, replace and repair are pretty similar. Like, are we replacing old cells? Are we repairing old cells? Are we replacing damaged cells? Whatever. According to your guide notes, we have 4: grow, replace, repair, reproduction. Okay? That's the 4 reasons why a cell is going to grow, or replace, or repair, or reproduce.
The order doesn't matter. So like you saw me write them in specific numbers. That doesn't matter. The only reason I did that was so we could put, so I remembered to put the G at the top. And GR might work for you, might not, whatever, but that's why. So I mean, if you had this order jumbled, there is no specificity to the order, okay? Just to clarify that.
4 phases of the cell cycle. We start with G1. What is the next phase? Now what goes on, let's talk about the next phase. After G1 comes
It's
S, and after S comes,
G2.
and after G2 comes
Mitosis.
M. M phase, which is mitosis. Yep, which is mitosis. Okay? G1, S, G2, M. Okay, you will know that M is mitosis. I don't care if you remember that G1 stands for GAP1. I don't care if you remember that S stands for synthesis. I don't care that G2 is GAP2. I don't care that you know what G and S and G's stand for. I do want you to know those in order, and I do want you to know the major events that occur during those phases. These 3 phases collectively, G1, S, G2, are all part of a process of the cell cycle that we collectively call interphase. An interphase is every part of the cell cycle where it's not dividing. Okay, where the cell physically is not dividing. So those 3 collectively we refer to as interphase. So during G1, the cell, one word, the cell grows. Absolutely, the cell grows. G1, the cell grows. It physically gets bigger. During S phase, what happens?
DNA is synthesized.
DNA is copied, DNA is synthesized, DNA is replicated, we make a copy. I don't care what word you want to put with DNA. DNA is copied, DNA is replicated. Whatever.
Why is that so blurry?
Okay, so that's what happens during S phase. During G2, a couple different things kind of happen. The cell prepares for division is often what we kind of generically say, and we often will say that organelles are replicating during G2. Now, in your POGL activity, the data table didn't match perfectly. Your hand is messing up the focus, I think, Mr. DeBoer. I'll figure it out. organelle doubling didn't happen during G2 according to that data table, but that's generally what we see in textbooks. And then during mitosis or M phase, what happens? The cell divides. The cell divides. And we make two genetically identical cells. During mitosis, the cell divides into two genetically identical cells. Now, after this, there is absolutely no reason why you do not have every single thing written down, because it's been put on the screen for you. And you say, well, what's the point of me copying it down? Mr. DeVore wrote it down. The point of copying it down is learning. Okay, you writing it down is another form of like, hey, G1 followed by S, followed by G2, followed by M. It's a way for you to interact with the learning.
Kinesthetic.
Aesthetically. To help you learn. Okay, if you say, well, I just sit there and absorb really well, I don't know if research suggests that. Some people, some people really do.
You're in the wrong phylum for that.
Okay, so the cell cycle, these are the main steps of the cell cycle, and these are the major events that happen in the cell cycle. We want you to know those.
Questions?
That is why we gave you this piece of paper, to try to see, one, warm up, do you know this yet? And if the answer is no, hopefully now this helps solidify it. So if, but I'm not saying we will, but if we were to give you this on Tuesday, you should be able to do it. I didn't plan on it. You should be able to.
Well, it's a good hint. If you get a blank piece of paper from a teacher and they say, do you know this, fill this out, do you think you might see it again in some sort of an assessment capacity?
Yes.
Uh-huh.
All right, so on this stool or on that lab table is a little piece of paper that you should grab one of. It also is linked via Canvas as a PDF copy. It's also linked via Canvas as a PDF copy if you would prefer to mark up a PDF. But the paper copies are on that stool or on that lab table, and I would strongly encourage for each and every one of you to grab a paper copy, unless you really, really like marking up PDFs. Okay, that's how I like to classify and use it.
Is this our new pet?
This is our new pet.
I thought it was like.
Well, we're just introduced, maybe on Tuesday, the actual puppy mitosis will come into the room. Maybe. We'll see. Maybe. I'll bring my dog in and pretend that her name's
Mitosis?
and that she's a puppy, not
Actually.
like a 12-year-old dog.
Exactly. You never say never.
So, we have a new puppy, new class pet. We're gonna call her Puppy Mitosis. Note the dog tag that has her name on it. She is young, and puppies need to get house trained, right? So what do you do so dogs don't pee all over? So they don't pee all over, thanks Mr. Ford. Pee all over the place. You use you let them outside, right? That's good, actually, yeah. You let them outside.
Or,
And you let them out regularly, and you reinforce it. Hey, good job, you went to the bathroom, here's a treat, positive reinforcement. However, when you're at school or at work and you can't be letting them out regularly, you might want to use a pee mat. Okay, you might want to use a pee mat, which is this little like absorbent thing that dogs can pee on, and then in theory it shouldn't get all over the floor. Okay? And pee mat is like this classic, I don't know, it's what most videos or textbooks, maybe not textbooks, but most people you talk to as a memory device, PMAT is kind of the classic go-to of the order of events in mitosis. It's a memory device. Maybe it works for you, maybe it doesn't. Maybe you want to come up with your own memory device. PMAT is kind of the one we'll talk about. Hey, our puppy mitosis, she's young, she's a puppy, so she needs a PMAT, so she doesn't go all over the floor.
Mr. DeGrand has
Someone told him, pass me a taco, as the order of events. Maybe you like tacos, and be like, hey, pass me a taco. And it's peanut, right? Pass me a taco. Okay, but these are the events of mitosis, and the order in which they occur. And we want you to know these, and we want you to know the major events that occur during these. So we have shown Amoeba Sisters a couple different times. This video that we're going to use today for Amoeba Sisters, like the other day, we used it to reinforce what the cell cycle and what we had taught. Today, we're going to use it to introduce mitosis. So during this video, they will talk about what the letters PMAT stand for. And while the video is playing, you should fill out what PMAT stands for. Okay? If we remember, we'll hit pause when they go through it. If they're going to go through and they're going to talk what all four mean, but then throughout the video they'll come back. They're like, oh, well, the P stands for blah, blah, blah. And they'll talk about what the P does. And during the M, you know, so if you don't get it the first time, they will come back. They will come.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes, very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for
Telophase. There is a P to the far left. It does say prophase, not prophase. And there is an E to the far right. It says telophase, not telophase. Okay? Prophase, metaphase, anaphase, telophase. Mr. DeBoer finishes so you can see how to spell it. You should be good. We're good, Mr. DeBoer?
Yeah.
All right.
telophase.
These are
So
coming back
remember
again, too.
PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, so PMAT, prophase, metaphase, anaphase, telophase. described on the way in. We want to go through and we want to talk about a few terms. So they referenced it in the video. They mentioned this thing called a chromosome. What the heck is a chromosome?
It's like DNA, but like together.
Yeah, absolutely. It's condensed like DNA. So if we look at a picture of what a chromosome is and kind of what it is, well, let's take a step back. What's DNA? I think we all kind of have a rough idea what DNA is, but what is it? Let's just kind of get that out of the way. What does it do? Why is it important? Yeah.
Passes down like information, like when it splits and passes on information, like what task a cell is supposed to do.
Yeah, really good. Yeah, it contains information and it tells what the cells to do. So if it's a skin cell, it's telling your skin cells what to do, like how to look, how to act. Okay, if it's a muscle cell, it's telling your muscle cells how to look. When I say look, I'm saying like how to form, like what it should physically look like, and what to do. If we're talking about a heart cell, well, that's technically a muscle cell. A bone cell, what it should do, what it should look like. Now, what's true about the DNA in your skin cells compared to the DNA found in your blood? Is it the same DNA or is it different DNA? Is it genetically identical, your blood to your skin, or are they genetically different? What do you think? Maybe think movies, TV shows. There's a blood splatter that's not the victim's. And we can extract some DNA from that blood sample. And if I think Mr. DeBoer was the person whose blood was left at the scene of the crime, do I physically have to go and suck blood out of his vein in order to get his DNA?
No.
No, what else could I do?
Oh wait, no. Yeah.
No. Fingerprints don't contain DNA. They are a physical imprint like of oils.
Okay.
But it is unique to Mr. DeBoer's fingerprint, and my fingerprints are unique.
But we're talking DNA.
But yeah, we're talking, it is something they use, but not genetic. Yeah.
Isn't it a swab or something?
A swab of,
The saliva?
yeah, absolutely. They sometimes take like a cheek swab or a saliva swab. Or maybe they could do hair. But yeah, so with that said, if Mr. DeBoer, we think Mr. DeBoer's blood was at the scene of the crime, we don't have to pull the blood from Mr. DeBoer to match the DNA. We could do a cheek swab. Okay, so what does that tell us? That the DNA in almost all of our cells is the same. It's genetically identical to one another. Because during mitosis, one cell divides into two genetically identical cells. And we are here today because egg and a sperm combined to make one fertilized cell, and then mitosis, 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 and each time genetically identical cells. Okay, so almost every cell in your body has the exact same DNA as the next cell. They look different because different parts of the DNA tell the cells what to do, depending on where they are, form versus function. And I say almost because egg and sperm cells don't. They have half, right? That's the sexual reproduction. So DNA, genetic information, is then packed together and is really, 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 really packed together to form a chromosome. So when we think about a chromosome, it's this traditional like X-shaped thing, and we can actually see them. If we were to find some cells that are going through mitosis under our microscopes, we can actually see the chromosomes. We can see what a condensed chromosome looks like. Okay? So when we talk about chromosomes, we want you to know the parts of a chromosome. So on that big piece of paper, okay, the one that you did as your warm-up on the way in, we're kind of right in the middle of that, we're going to ask you to draw a chromosome. Okay, so Mr. DeBoer's drawing a chromosome for us, and when we talk about chromosomes, it's this like typical, it's an X. Now, when I use my hands I'll use my hands a fair amount of times to say, hey, this is a chromosome. I will put my hands together like this, where my two X's join at my wrist, not like this. Okay, just like Mr. DeBoer, someone in my 6-hour class, so it kind of looks like two boomerangs. Yeah, it's kind of like two boomerangs put together. Okay, and each hand is a boomerang, okay? And what will happen during mitosis is the hands, or my boomerangs, or my X's will separate, they'll pull apart, okay? And they're joined in the middle by this protein, and that protein is called a centromere. How many chromosomes did Amita sister say you have, or do you know how many chromosomes you have in like your skin cells, your hair cells, your muscle cells, your bone cells? 46. You have 46 chromosomes. How many pairs is that? Well, a pair is 2, so 46 divided by 2 is?
96.
Divided by?
96.
23 pairs. Good. We're 23 pairs. So we have 46 chromosomes. Divide by 2, we have 23 pairs of chromosomes. And when we talk about pairs of chromosomes, here is this poster is what we call the human genome. So they've actually gone through and they've traced what each part of each chromosome is responsible for, and they have a list, and it's written really, really small. But if you want to look at it sometime, you're more than welcome to. So, but there's 23 pairs, and that's what these numbers represent. Now, you have 23 pairs of chromosomes. Where did half of those chromosomes come from? Mom.
Uh-uh.
Okay? And where did the other 23 come from?
Yeah.
Dad. Birds and the bees. Okay? A sperm cell combined with an egg cell, 23 plus 23, 46. You are now 46 chromosomes. You're a single cell that divides and divides and divides and divides. 37.2 trillion cells are in your body as an adult about. But it all started from one fluorescent. Okay? So a chromosome, when we talk about pairs of chromosomes, and that's on this poster, they're numbered. So we have 2 chromosomes, number 1. We have a pair of chromosome number 1. One came from your mom, one came from your dad. You have a pair of chromosome number 2. One came from your dad, and one came from your mom. You have a pair of chromosome number 5. One from dad, and so forth. Collectively, this X, the 2 boomerangs put together in the middle by a centromere, it's a protein called a centromere, is called a chromosome. Collectively, we call that a chromosome. Each half of the cell is called a chromatid. Chromatid. Now, in your notes, we've talked about chromatin with an N. This is chromatid with a D. Chromatin is uncoiled DNA. For chromatid is condensed. I don't know. Besides the D. Condensed DNA. A chromatid. We have 2 chromatids when we talk about a chromosome. Collectively, we call each chromatid sister chromatids. Okay? They are sister chromatids to one another. And sister chromatids are genetically identical to each other. Now, why are they genetically identical? Because during S phase, what happened?
DNA copy.
Copy, right. The DNA got copied. So one side is a complete set of DNA, and the other side is a complete set of DNA. Now, through the process of mitosis, those sister chromatids will pull apart. And then eventually each sister chromatid will be in its own cell. But if one sister chromatid is genetically identical to the other sister chromatid, final end result is we have 2 cells that have a complete set of DNA because each sister chromatid is genetically identical. Now, how many pairs of chromosomes do we have? We have how many pairs of chromosomes?
46.
How many pairs of
Two.
chromosomes?
23.
23 pairs because one came from your mom and one came from your dad. Good. Okay. We have 23 pairs of chromosomes. So I'm going to make a number up. That's chromosome pair number 1. The one on the left came from your mom, the one on the left came from your dad. I'm making that up, right? Who knows? It could be flipped. It doesn't matter. But one of those came from mom, one of those came from dad. They are a pair of chromosomes. Mom's chromosome number 1 codes for certain traits. Dad's chromosome codes for the same, hang on, let me rephrase it. There are these sections, let me take a step back, actually. On the chromosomes, so these are called homologous pairs. These 2 chromosomes are homologous pairs. So this is one came from mom, one came from dad, they're homologous pairs. Homo means same, homosexual, heterosexual, think of those prefixes. Homo means the same, people prefer people who, same gender, homo, or heterosexual, different. So homologous, homo means same. These are the same pair of chromosomes. A pair of chromosomes is chromosome number 1 and chromosome number 1. Okay? On these chromosomes, there are these bands or sections that contain something called a gene. And we call them genes. So what a gene is a gene is a section, a location, a segment of DNA that codes for a particular trait. Okay, so we often think of gene as like a location, but it's a chunk of DNA that's responsible for a particular trait. Example would be hair color. On chromosome, I'm lying right now, on chromosome number 1, that orange band is hair color. And the one on the left came from mom and the one on the right came from dad, chromosome-wise. So they have the exact same genes. Homologous pairs have identical genes. Hair color, hair color. Okay, hair color is the gene, it's what we are, what the trait is. However, they might have different variations of the gene. A gene, again, is a location, a section of DNA that codes for a particular trait. So this might be hair color. Is that what I've been saying? I think I've been saying hair color. Okay? Now, those bands on the bottom, that might be eye color. Both homologous pairs have the same gene. They have the same location that codes for hair color, or they have the same section that codes for eye color. Okay? Those are the same genes. However, does mom necessarily have the same eye color as dad? They both have eye color, they both have hair color, they might have different variations of hair color. So those orange bands, both are hair color, but mom maybe has blonde hair, and dad maybe has brown hair. Okay? The bands on the bottom, mom might have blue eyes, and dad might have brown eyes. But they're both eye color. Now this is when we talk about genetic traits, we often very oversimplify it. We kind of pretend like there's one gene that controls one trait, where if you were to actually look at this, hair color is on here lots of different times. There's often a lot of different genes that combine to make your hair color.
What if there's a mutation like heterochromia?
If there's a mutation that what?
Like heterochromia, what would it look like?
A mutation for what?
Heterochromia.
Heterochromia? Chromia, what's that? Is that 2 different eye colors?
Yes.
I don't know. I don't know the answer. So somewhere along the line, there was probably some type of mutation, and that type of mutation got passed on. Why? I don't know. Something went wrong with the copying of the DNA. Okay? Or the combination of certain traits then gave you the traits that trait showed up in. Okay? So when we talk about chromosomes, we talk about genes, we talk about homologous pairs, we talk about sister chromatids. Homologous pairs are different because one came from mom and one came from dad. Sister chromatids are genetically identical because sister chromatids were replicated or were made during S phase. Okay? Sounds like we're getting close. Looks like we're getting close.
Wait, oh wait, no, never mind, I'm thinking about cells, I'm thinking about daughter cells. Well, you talked about sister chromatids. So confused.
Okay, so sister chromatids are copies of DNA.
Yeah,
Daughter cells are the final product of mitosis.
that's, yeah.
Yep, and they are also genetically identical.
Okay.
Okay, so on this stool or on that chair is your homework for by Tuesday. Guided notes, also linked as a Google Doc and as a PDF via Canvas. The video is uploaded, the Google Slides presentation is uploaded. If you have any questions, it's in Canvas, but let us know, email us, and we can help you out along the way. Have a great weekend, everyone. We will kind of finish this up on Tuesday or Monday.